Hi, welcome to this next video tutorial in our general biology course. This session is entitled Meiosis. So in our previous videos, we talked about a process called mitosis. That's used for growth and repair, simply making more identical cells. In this video, we want to talk about a process of cell division that can be used for sexual reproduction. For example, the production of sperm and egg cells. So let's take a look at our learning objectives. We want to make a distinction between sexual and asexual reproduction. So reproduction that involves combining genetic material from two parents, that would be sexual, or asexual, where one organism can simply clone itself. We'll want to talk about a process of cell division called meiosis. As I just mentioned, this is the type of cell division used to produce gametes, which is just another name for sperm and egg cells. And we'll see how meiosis can lead to genetic diversity. And that's important in biology, particularly as we move on later in the semester into the topic of evolution. Evolutionary change requires a constant renewal of genetic diversity. And then finally, we'll talk about non-disjunctions. This is something that can happen when meiosis doesn't work correctly, and what some of the consequences of that are. So that's where we need to go. So let me remind you one more time of mitosis. This is the process by which eukaryotic cells multiply simply to produce more identical cells. We've been through this a couple times now. So a cell is in interphase when it's not undergoing cell division, but at some point during interphase, specifically S of the cell cycle, it will duplicate its chromosomes. And now it is ready to enter mitosis. So prophase, that's when the chromatin condenses into chromosomes and those spindle fibers begin to form. Prometaphase, the spindle fibers attach to the chromosomes and the nuclear envelope breaks down. In metaphase, the spindle fibers are already starting to tug and shorten a little bit. That pulls all the chromosomes to the middle, and notice that they line up single file. So I see 4 chromosomes there in metaphase, each one existing as duplicate sister chromatid copies. And then in anaphase, the spindle fibers truly begin to shorten, pulling those sister chromatids apart, and at that point we call them chromosomes in their own right. They move towards opposite poles of the cell, and then finally in telophase, the chromosomes arrive, new nuclear envelopes form, cytokinesis begins. That's when the cell splits in half. So mitosis. Mitosis is when a parent cell divides to produce 2 genetically identical daughter cells. And we have said that this is used for growth and repair. So if you fall and scrape your knee and you need to replace that skin, we can use mitosis just to make more cells to patch that up. Or growth, you began as a fertilized egg cell, but that fertilized egg cell had to go through many rounds of cell division to produce an embryo and a fetus and a baby and eventually an adult. That's all mitosis. But mitosis can also be used for asexual reproduction. In fact, that's what this organism in the picture is doing there. That green thing is called a hydra. It's about 1/8 of an inch tall. You can almost literally see some of the cells that form it, particularly around the edge of it where they're more white in color. You can almost sort of make out cell boundaries. But the idea is this adult organism, this parent organism, we're not necessarily meaning just parent cell now, we mean a parent organism, can clone itself asexually, make more of itself simply by using mitosis. In fact, you see that little bud growing off the side, that little mini-me, it's just cloning itself. It's making an exact copy of itself by dividing its own cells via mitosis. So that's a form of reproduction, and it works fine. It's just the offspring will be genetic clones of the original parent. And we're going to get into a process in this video here where reproduction can occur sexually. So if you take a look at this picture, we see 2 parents, the father on the left, the mother on the right, and in the middle you can see their 3 kids. And notice that none of those kids are clones of the parent. They're not even clones of each other. They all look different. And so how did we create genetically distinct offspring, very different from what that hydra is doing? Well, it requires a different type of cell division called meiosis. Sounds a lot like mitosis, but meiosis is a very different process. So to get started thinking about how this works, let's remind ourselves that humans have 46 total chromosomes in their cells. So whether we're talking about the mother or the father or any of those kids, if we looked inside their cells, we would find 46 chromosomes. So think about that child in the middle. She's got 46 chromosomes in every single one of her cells, but where did she get her 46 chromosomes? Well, her father contributed a sperm cell that had 23. Her mother contributed an egg cell that had 23. And when the sperm fertilized the egg, that produced a fertilized egg, a zygote, that had 46 total chromosomes. So that became the starting cell from which all of her cells developed. But how did we get a sperm cell with 23 and an egg cell with 23 if I'm telling you that human cells have 46? Well, sperm cells and egg cells are unique. They're referred to as gametes, and they have half the number of chromosomes, and they get them because they are produced by a type of cell division called meiosis. Now, once the sperm and the egg fertilize each other and we get our 46 chromosomes in the fertilized egg can now go through cell divisions and make more cells and more cells and more cells. But it's going to use mitosis at this point, so all of those cells will continue to have 46. We're just going to be cloning the cells over and over and over to get this ball of cells called a blastula. And more cell division, more mitosis, growth, more growth. We get this little fetus growing now, and more cell division and more cell division. And eventually, through mitosis, we'll get this full-sized baby. Every cell in this baby's body has 46 chromosomes, derived from the original 23 that came from the sperm and the 23 that came from the mother's egg. So meiosis is used to make the gametes. Mitosis is then used to create the baby once the gametes have fused to restore the number of 46. And eventually the baby is born. And if we looked at the chromosomes inside one of these baby's cells, let's just take one of the cells out of this baby and look at its chromosomes, we would see a picture like this. And this is under an electron microscope, but this is called a karyotype, a human karyotype. And you are looking at all 46 chromosomes there. But notice that they come in pairs, so they've numbered them. Pair number 1 is shown in that white box there. So there are 23 pairs. I know you only see numbers up to 22 down there at the bottom. The next pair says X and Y. That 23rd pair are the sex chromosomes that determine the sex of the offspring. And if you have an X and a Y, that would be a male. If you happen to have 2 copies of the X chromosome, that would be a female. So that 23rd pair, the sex chromosomes, determine the sex of the offspring. The other 22 pairs are called autosomes. So humans have 23 pairs of what we call homologous chromosomes. Homologous literally means same word. So what it's trying to imply there, if you look at pair number 1 in the white box, one of those chromosomes came from the sperm of the father, the other came from the egg of the mother, but they're homologous, same word, meaning they carry essentially the same genetic information. For example, the father will give a chromosome with a gene on it for how to make hemoglobin in blood, and the mother's egg will contribute a chromosome with a gene on it for making the hemoglobin in blood, for example. So they're homologous pairs. They're carrying essentially the same genetic information. Now we know the genetic information is not completely identical. We'll get into this more as we talk about genetics in a future video. So humans have 23 homologous pairs, essentially a set of 23 from each parent. So the offspring receives 23 chromosomes from the sperm, 23 from the egg. That gives us 23 homologous pairs and 46 total. So in other words, the gametes, the sperm and egg cells, gametes are haploid because they only have one set of chromosomes. They have one set of 23. 1N is the way that's often written, in meaning number of sets. They have one. So haploid means one set of chromosomes. In the case of human cells, haploid would mean 23. And the offspring, however, are diploid because they get a set from each gamete. So they are diploid. They have two sets, 2N. One set from the sperm, one set from the egg. And then once we get that diploid fertilized egg cell, it can undergo many rounds of cell division via mitosis to produce more diploid cells just like it. So take a look at this figure for a second. Down there in the bottom left, we see this couple that are going to produce a fertilized egg. So the mother, the wife on the left, has ovaries. Ovaries are where the eggs are produced by meiosis. So the cells in her ovaries begin with 46 chromosomes, with the 23 homologous pairs. But using meiosis, her egg cells only get one set of the 23, so her egg cells are haploid. Meanwhile, next door there, the father in the testes produces sperm cells using meiosis, and those sperm cells are also haploid. The sperm cells only have one set of 23. The sperm and egg then fuse and fertilize during the act of fertilization, so we now get a fertilized egg, also called a zygote, that gets a set from each, so it now has two sets, or a total of 46. And that fertilized diploid zygote can now undergo mitosis and development, producing more cells with 46, more diploid cells, as it grows into an adult, and the cycle just continues over and over. So this allows for sexual reproduction. We can combine genetic information from two different parents so long as the parents only contribute half the typical amount of genetic information. And again, the gametes, the sperm and egg cells, are produced by a type of cell division called meiosis. So let's look at meiosis in a little bit more detail. And we're going to simplify it at first. Humans, I know I've said, have 46 chromosomes or 23 homologous pairs. Let's look at a cell right now that only has 2 chromosomes or 1 homologous pair. So the cell you see there has, I see 2 chromosomes there, the blue one and the red one, so there's 2 chromosomes, but they represent a homologous pair. Let's assume the blue one originally came from the father, the red one originally came from the mother, so we have a pair. If we're going to go through meiosis, the cell will start in interphase, very similar to mitosis, and then it will actually go through 2 cell divisions, meiosis 1 and meiosis 2. They're similar to mitosis, but I'm going to try to point out some key differences, which results in these haploid gamete cells. So the cell is in interphase, but as we've said before, during interphase, that's when cells will duplicate or copy their genetic information. So notice the 2 chromosomes have each copied themselves. So in the cell we're looking at now, I still see 2 chromosomes, the red one and the blue one, but each chromosome now exists as duplicate sister chromatids. So in that cell, I see 1 homologous pair, I see 2 chromosomes, the red one and the blue one, and I see a total of 4 sister chromatids because each chromosome has been duplicated. This cell can now begin the first round of cell division, which we're calling meiosis 1. And what's going to happen is during metaphase and anaphase, the homologous pair is going to find itself in the middle. So rather than line those 2 chromosomes up single file, they're actually paired next to each other. So as we go through meiosis 1, the homologous pairs separate. We see the blue member of the pair going to the bottom. We see the red member of the pair going to the top. So technically, we've already created haploid cells because haploid cells would only have 1 chromosome. The original cell way back over there on the far left has 2 chromosomes. The cells we've generated now through meiosis 1 each only have 1 chromosome. Granted, each chromosome exists with its duplicate copy, its sister chromatid. In fact, that's why we need meiosis 2. The purpose of meiosis 2 now is to separate the sister chromatids. So again, we can put the chromosome in the middle of the cell, throw out ropes, and pull apart the sister chromatids, and now we'll end up with 4 cells, 4 haploid cells. So remember, go back to the very far left over there. That cell has 2 chromosomes, the red and the blue, 1 homologous pair. But that pair is going to separate during meiosis 1. Red went to one side, blue to the other. And then we'll separate the duplicate sister chromatids in the second meiotic division. So we're going to get 4 haploid cells. Think of that as 4 sperm cells or 4 egg cells, however you want to think of it. But we end up with 4 haploid cells after undergoing meiosis 1 and 2. Now that was a simplified diagram because we were only considering one homologous pair, just to get the big picture, just to see what's going on. Let's ramp things up a bit and let's look at a cell that has more chromosomes and see if we can keep track of everything that's going to take place here. So once again, I see this thing right here. That is a homologous pair. I can tell it's a pair because there's a red, there's a blue, and they're of the same size. So this is like the medium one. If you notice, there's a small pair and a big pair in there as well. So that's a pair of homologous chromosomes. Remember, homologous chromosomes are a pair of chromosomes that carry similar genetic information. One originally from the mother, one originally from the father. Think red from the mother, blue from the father if you want. So there is a homologous pair. So I see three homologous pairs. I see the small red and blue pair, I see the medium red and blue pair, and I see the large red and blue pair. So there are three homologous pairs for a total of 6 chromosomes. This is a chromosome right there. That's the big blue chromosome. And I see 6 chromosomes. I see big blue, big red, medium red, little blue, little red. So there are 6 total chromosomes, 3 homologous pairs. And clearly this cell has already gone through the process of duplicating its chromosomes because that chromosome is existing as 2 duplicate copies, 2 duplicate sister chromatids. So in this cell I see a total of 12 sister chromatids. So let's just be sure you're keeping these terms straight. This cell has a total of 6 chromosomes, much like your cells have a total of 46. It has 3 homologous pairs, much like your cells have 23 homologous pairs. And it has 12 sister chromatids because it's already gone through that step of duplicating or copying its chromosomes. So hopefully all that's clear. So now let's see what happens as it enters into meiosis 1. So we're starting out in interphase. That's when the chromosomes also get duplicated. In the picture I just showed you, they have been duplicated. So we're going to now leave interphase with our duplicated chromosomes and enter the first meiotic division, which is called meiosis 1. That's the meiotic division when the homologous pairs are going to separate from each other. Now we're going to see all the same features that we saw in mitosis. So we've got prophase, metaphase, anaphase, telophase, cytokinesis. But this is meiosis, specifically meiosis 1, so we're calling it prophase 1, metaphase 1, anaphase 1, and telophase 1. Now prophase 1 is exactly like it was in mitosis. So in prophase 1, the chromatin, the tangled mess of all the chromosomes, begins to condense into visible chromosomes, and the nuclear envelope begins to break down because we're not showing a pro-metaphase here. And the spindle fibers are starting to form. So this is exactly like we had seen in mitosis. Now I want to go ahead and alert you to something interesting that's taking place. Let's not worry about the consequences of it yet. We'll talk about that later. But do you notice how the homologous pairs, the big, medium, and small pair, have already kind of found each other? They found their mate, and they've even flopped over each other a little bit. They're doing something called crossing over. And as we'll see here in a second, when they cross and flop over, they will exchange tips of their chromosomes. And the significance of that, we'll talk about that more later, but I'm just alerting it to you now that during prophase 1, this crossing over process occurs, where the sister chromatids will actually exchange tips with the homologous sister chromatid. All right, so now here is where things really get different. It's in metaphase 1. This is where meiosis really differs from mitosis. In metaphase 1, notice that the homologous pairs get next to each other. If this were mitosis, they would all be lined up single file, and then we'd pull chromatids to opposite ends. But in metaphase 1 of meiosis 1, the homologous pairs actually get next to each other in the middle. We've thrown the ropes out, the microtubules, they've latched onto the kinetochores. So a lot of it's just like mitosis, but it's this finding your homologous pair and getting next to it that differs. Now, during anaphase 1, what we're going to pull apart are the homologous pairs. So you see big red going to one end and big blue going to the other end. Same with the other pairs. And notice the colored tips that are different. So we'll talk about that more later. But what we're separating in meiosis 1 are the homologous pairs. So by the time we get to telophase 1 in cytokinesis, we essentially only have one set of chromosomes. Go back and look at the cell in prophase 1. I see a total of 6 chromosomes. 3 homologous pairs, the big pair, medium pair, small pair. What do we got? 12 sister chromatids. But the important thing is 3 chromosomes in the cells that result at the end. We get a big, a medium, and a small in each of those daughter cells. So go back to telophase there. You see a big, a medium, and a small in each daughter cell. Granted, they're still in their duplicate chromatids, but there are 3 chromosomes in each of those cells, a big, a medium, and a small, whereas the original cell back there in prophase 1 had 2 bigs, 2 mediums, 2 smalls, had 2 sets. These cells out of telophase 1 only have 1 set. In fact, here they are. Here's the cells we get out of telophase 1. They're already haploid. The diploid cell would have had 6. These cells only have 3, a big, a medium, and a small. So we've already got our 2 haploid cells. But because they still exist with their duplicate chromatids, we have to go through a second meiotic division to get those chromatids apart. So that's called meiosis 2. That's when the sister chromatids separate. It consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. So prophase 2, same old story. Chromatin condenses into chromosomes and spindle fibers form. Now in metaphase 2, notice what happens. The chromosomes do line up single file. They can't get with their homologous pair because their homologous pair's over in the other cell at this point. So they line up single file. We've thrown out the microtubules. They've latched onto the kinetochores, and they will begin to shorten and pull the sister chromatids apart. And they'll eventually arrive at opposite poles, and we'll end up with our 4 haploid cells. Now notice all 4 cells will be haploid because they will have 1 set. They'll have a set means a big, a medium, and a small. And each of those 4 cells will have a big, a medium, and a small. So they'll have a representative of each homologous pair. Those 4 cells will become sperm cells if we're talking about a male, or those 4 cells will become egg cells if this has taken place in a female. Now, we've only shown a cell that started with 6 and haploid became 3. Remember, your cells have 46 and haploid would be 23. So we go back to this. We've got a sperm cell with 23, we've got an egg cell with 23, and they fertilize each other, and that produces a diploid fertilized egg cell with 46. We're getting a set from the sperm and a set from the egg. So when does meiosis take place in humans? Well, in males, this process of meiosis, this process of making gametes, males begin producing sperm via meiosis at puberty. So when a male turns, that varies, but let's say roughly 11 or 12 years old, they enter puberty, their body starts producing testosterone for the first time, and testosterone is what stimulates meiosis in the testes to produce sperm cells. So a male begins making sperm by meiosis at puberty, literally making hundreds of thousands or millions a day. And the male will continue this sperm production 24/7, 365 for the rest of their life. So you guys who are watching this video right now, you are actually multitasking. You're concentrating and taking notes and answering study guide questions, but you're also cranking out thousands of sperm. So 24/7, 365 for the rest of their lives, that's when males make sperm cells. They make a whole bunch more every single day if the sperm, and they're stored in the testes. If they're not used or needed, they will be dissolved after a few days, and new ones are constantly being made to replenish those. So that's males. Females are a bit different. So females begin producing eggs via meiosis while they're in the uterus of their mother. So you females watching this video, you're done with your meiosis. You did all that while you were still in the uterus of your mother. Your ovaries produced all the eggs by meiosis. So what's happening at puberty? Well, at puberty, again, age 12-ish, females begin ovulating an egg. That's different than producing the egg by meiosis. Ovulating means they release an egg out of the ovary into a tube called the oviduct and let the egg sit there to see if it becomes fertilized. Obviously, way more times than not, it is not fertilized. So the egg just dissolves, and then 28 days later, another egg is released. So females begin releasing an egg at puberty, but they've done all their meiosis while they were still in their mother's uterus. And this ovulation of an egg continues until the female reaches menopause, which can vary tremendously, but roughly around age 40. Some women reach it earlier, some women a little later, but on average about age 40. So there's a little difference between when meiosis is occurring in males and females. Now here's a figure that I think is a great summary comparing mitosis to meiosis. So it's two different types of cell division. Remember the purpose of mitosis, growth and repair. We just want more identical cells. Whereas meiosis, we want to make haploid cells with one set of chromosomes. So let's take a cell right here. This is a diploid cell. It's got 6 total chromosomes. So it has 2 sets of 3, basically. A set consists of a large, medium, and a small. It's got 2 sets. It's got a pair of large, a pair of medium, and a pair of small. So this could be the starting cell, whether we're talking about mitosis or meiosis. So let's just see what would happen if we went the mitosis route here. To begin mitosis, the first step is to duplicate or copy the chromosomes. So I still see 6 total chromosomes, 3 homologous pairs, but they've all now duplicated, so they exist as sister chromatids there. 12 sister chromatids in those cells. This is exactly what would happen before meiosis begins. We would copy the chromosomes, so I still see 6 total chromosomes, 3 homologous pairs, 12 sister chromatids. Let's go back to mitosis and see what happens. In mitosis, the chromosomes will all line up single file in the middle of the cell, and we'll throw the ropes out, latch onto the sister chromatids, and then start pulling sister chromatids apart. If we were talking about meiosis, specifically meiosis 1, notice what happens during metaphase of meiosis 1. The homologous pairs are not single file. The homologous pairs are actually next to each other. So as we throw out those microtubule ropes and start pulling, we're going to pull homologous pairs apart in meiosis 1, whereas over there in mitosis we were pulling apart sister chromatids. So we go back over there to mitosis, we pull the sister chromatids apart, and we've now produced 2 daughter cells that are also diploid. They're exactly like the original parent cell. Remember, mitosis just starts with a diploid cell and makes 2 identical diploid cells. So I see again down there at the bottom 6 total chromosomes, 3 homologous pairs. The 2 cells are identical to each other and they're exactly like the original parent cell. But let's go back over to meiosis. In metaphase 1, we paired up the homologous pairs, so that's what we pull apart, and we've now got haploid cells. Each of those cells has a big, a medium, and a small. A big, a medium, and a small in each cell. But they exist as sister chromatids still, so we do our second meiotic division, and that produces our 4 final haploid cells, the 4 sperm cells or the 4 egg cells, depending upon if we're in a male or a female. And I want us to now start going back to this idea of creating genetic diversity. If you look at the 4 haploid cells down there, you can already see some differences. Those 4 cells don't even look alike, and that was just dealing with a few chromosomes here. So let's start talking a little bit more about this genetic diversity that we can create using gametes and sexual reproduction. If we go back to this family, the father on the left, mother on the right, kids in the middle, I pointed out that the offspring are not clones of the parents. Nature has created some new genetic combinations in the offspring. And again, as we'll talk about later this semester in our evolution unit, evolution relies on this genetic diversity. We need constant genetic change to drive the evolutionary process. And meiosis and sexual reproduction can do this. In fact, meiosis and sexual reproduction create genetic diversity by 3 different ways, or at least 3, but 3 that we're going to talk about. The 3 ways that meiosis can create this genetic diversity, well, we get that crossing over during prophase 1, so we're going to come back to that, where those chromosomes flopped over each other. We're also going to see something called random assortment of homologous pairs during metaphase 1 and anaphase 1. And then just random mating events, which 2 parents actually find each other and have the offspring. There's all sorts of combinations there. So let's start with this crossing over during prophase 1. Let's go back to meiosis 1 and take a look at that again. So remember, in meiosis 1, we start with a cell that's in interphase. It copies its chromosomes, and then that cell enters prophase 1 of meiosis 1. The chromatin condenses into visible chromosomes, so we can see the actual chromosomes, but the homologous pairs have already found each other. You can see the big pairs next to each other, the medium pairs next to each other, the small pairs next to each other, and they're flopping over in what we're calling crossing over. And as I mentioned, they'll swap tips. Sister chromatids of opposite pair there will swap tips, and let's see how that would play out. Again, think of the red chromosome as containing all the mother's genetic information, the blue, if that came from the sperm cell, containing all the father's genetic information. And duplicate sister chromatids would still have the same genetic information. The 2 big reds are identical. The 2 big blues are identical to each other. But if we start swapping tips, we're going to create new combinations right away. So take a look at this figure here. It's taking us from prophase 1 to anaphase 1 to anaphase 2 to telophase 2. And let's look right there. So there's where the crossing over is occurring, and they're only showing it for 1 homologous pair, the big pair. But look beneath it now. We've created completely different types of chromatids. Without crossing over, we should see 2 big blues on the left and 2 big reds on the right. But this crossing over has created hybrids, combinations of maternal and paternal genes already on different chromatids. And so you see how we now enter meiosis 2 and get the chromatids apart. And at the bottom, you see that out of the 4 gametes that are produced, they all have a big chromosome. They all have 1 member of the big pair, but none of them are identical. And that's just for the big pair. In your cells, you got 23 pairs going through this. So we're already seeing some new combinations of genes in the gametes. No 2 sperm cells are going to be exactly alike. No 2 egg cells are going to be exactly alike, even when produced by the same person. The second way we create some genetic diversity is through the random assortment of homologous pairs during metaphase 1 and anaphase 1. So let's look at that. So here's a cell. This one happens to have 2 homologous pairs, so a little more complicated than the last picture. Has a big pair and a small pair. And during metaphase 1, when the homologous pairs get next to each other, they could get next to each other like this. So it just happens that the blue ones got on the left and the red ones got on the right, but the big pairs together and the small pairs together. It's just as likely that they could have lined up like this, the big pairs together, small pairs together, but notice that the red and the blue are not necessarily on the same sides as they were. In other words, it is completely random as to which side they get on. For any pair that gets in there, the blue could be on the left or the right. For the next pair, the blue could be on the left or the right. And we got 23 pairs. They're only showing you 2 pairs here. So then, when they move apart during meiosis 1, you end up with cells like this. And then in meiosis 2, here's what you'd get. You look down there at the bottom, and you see we get a big blue and a little blue, a big blue and a little blue, then a big red, little red, big red, little red. That's if they lined up in that orientation. If they had chosen the other orientation, here's what we get. We get a big blue, little red, and then a big red, little blue, big red, little blue. So notice once again that the gametes are not identical to each other. And this doesn't even show the tip crossing that we already talked about. So again, the point is we're getting unique combinations of genes in these gametes. A mother, no two egg cells of hers are alike. A father, no two sperm cells of his are alike. And then finally, we get genetic diversity simply by the random mating events. Who is the father? Who is the mother? That we know no two sperm cells from a father are alike. No two egg cells from a mother are alike. So that's already going to be random enough. But now you're making it random essentially as to who the source of the sperm is and who the source of the eggs are. In other words, anytime a child is born, it is an unbelievably unique new genetic combination that's never before ever been on Earth in the history of life. And again, we'll see that's the driving engine of evolutionary change, is creating genetic diversity. Now, let's look at this karyotype for a second. Remember, I showed you a karyotype earlier in the video that showed the 23 homologous pairs. Well, there's something very unique about this karyotype. If you look right there, there are 3 copies of pair 21, or 3 copies of chromosome 21. So it's not exactly a homologous pair. We got a homologous triplet right there. You're not supposed to have 3 copies of any of your chromosomes, nor are you supposed to only have 1 copy of any of them. They're all supposed to come in pairs. And if you think about it, this would be 3 copies of 21. That's called trisomy 21, which you may know better as Down syndrome. So Down syndrome is a genetic condition that results when a child has an extra copy of chromosome 21. It's possible that a fertilized egg could end up with multiple copies of other chromosomes. You could have 3 copies of number 12 or 3 copies of 10 or only 1 copy of 7 or something like that. But whenever the chromosome number is incorrect, it's almost always lethal. In other words, the fertilized egg doesn't really develop at all. It's spontaneously aborted. One of the few exceptions is trisomy 21. It is possible to have 3 copies of chromosome 21 and still produce an offspring. You can literally see there, just looking at 21, it's a very small chromosome, so it doesn't have as many genes as some of the others, and that contributes to the fact that you can tolerate this extra copy because there's just not as many genes to be messed up with here. There are consequences. There are effects of Down syndrome, and I hope sometime in your life you have or will meet someone with Down syndrome because they are fascinating people. They're usually just the happiest go lucky people you'll ever meet, very kind, very joyful. So although there are consequences, it still creates an extremely unique individual. So how did this happen though? Well, it results from what's called a non-disjunction. A non-disjunction is an incorrect separation of chromosomes during meiosis. So we know that the homologous pairs are separating, the sister chromatids are separating, but what would happen if something didn't go right and we had both copies of something going to one side and neither copy going to the other side? Well, that can result in extra or missing copies of chromosomes in the gametes. And if the gametes have the wrong number, well, then the fertilized egg is going to end up with the wrong number as well. And the non-disjunction, the incorrect separation, can occur during meiosis 1 or meiosis 2. So let's take a look at it. In the case of trisomy 21, what it means is either the sperm or the egg brought 2 copies. It should have only brought 1 copy, and it brought 2 copies. Combine that with the single copy from the other parent, you end up with a total of 3. So how could you get a sperm or an egg cell that has 2 copies? Well, let's first look at how this could happen during meiosis 1. So here is it playing out. So if you look up here, in meiosis 1, look what's happening right here. We have said that during meiosis 1, the homologous pairs separate, go to opposite ends of the cell. But notice what's happened is the pair that's in the box there, both copies are going to the left. Neither member of that pair is going to the right. So as you look at the cells beneath it now, we're going to get extra copies of the big chromosome. Since both homologous pairs went to one side, we'll then separate those sister chromatids in meiosis II, but notice down there at the bottom, you're getting some gametes with the incorrect number of chromosomes. We get 2 of them there with an extra copy of the big, and we get 2 gametes that are completely missing a copy of the big, all because of a non-disjunction during the first meiotic division. The homologous pairs didn't separate. But you can also get a non-disjunction during meiosis II, so let's look at how that would play out. So let's assume, you can see at the top there, that the homologous pairs did separate fine during meiosis I, and it was during meiosis II that the problem arose. So the sister chromatids are supposed to go to opposite ends of the cell, and we see there, the one in the box, the 2 sister chromatids did not separate, they both went to the left, and neither went to the right. Follow that down to the bottom, and you see once again we're creating gametes, either sperm or egg cells, that have missing or extra copies. Any of those that say +1 are gametes that conceivably could be causing Down syndrome if the chromosome that went through non-disjunction was chromosome number 21. In other words, if you got an extra copy of chromosome 21 in any of these gametes, they could then fertilize a regular gamete and produce the trisomy 21. Now, I showed you an example using trisomy 21 because nearly all the other chromosomes, if you get an extra or missing copies, it just doesn't work. The only exception to that are the sex chromosomes. Normally, everyone would have a pair of sex chromosomes. Females have an X and an X, males have an X and a Y. It is possible to have the wrong number of sex chromosomes. You could be XXY, XYY, or even an X with no Y. The only thing you can't be is just a Y chromosome. But having extra copies of the sex chromosomes actually works. The way you know what sex it is by whether there's a Y or not. If a Y chromosome is present, you have a male. Doesn't matter how many X's you have. So you can have non-disjunctions actually result in viable humans with sex chromosomes. But the one we hear about the most and talk about the most is the Down syndrome, the trisomy 21. All right, well, we've made it through our 3 lectures on cell division. And our next one, I believe, we'll start talking about genetics. So we'll see you in the next video.